Hello friends, we are going to present about PCR and RT-PCR. In the technical world, there are many techniques used to identify the new subjects. Therefore, in our visual art, we focused on PCR and RT-PCR. Using PCR, millions of copies of a section of DNA are made in just for few hours. There are many important things we need during PCR process. Primers are small pieces of DNA that are made in a laboratory. These are necessary for PCR process because DNA polymerase can attach to any old place and start copying away. It can only add on to an existing piece of DNA. DNA polymerase is a naturally occurring complex of proteins whose function is to copy a cell's DNA before it divides into two. Nucleotides, they are the building blocks. The DNA molecules are made up of 4 types of nucleotides in PCR reactions: adenine, cytosine, guanine, and thymine. There are many techniques involved in PCR process. Step 1. Heat separate double-stranded DNA into single-stranded DNA, since hydrogen bonds linkage bases to one another are weak. They break higher temperature where the bonds linkage deoxyribose, phosphate, which are strongly covalent bonds. Step 2. Annealing usually takes place between 42 to 60 degrees Celsius depending on the length sequence of primers. These allow the primer at annealing to specifically target sequence. Step 3. Once the primers of annealing to the complementary DNA sequence, the temperature is raised approximately 72 degrees Celsius and thermostable polymers begin to synthesize new double-stranded molecules which are identical to the original target DNA. End of the cycle. At the end of the first PCR cycle, there are now 2 new DNA strands identical to the original target. However, that the DNA polymerase does not recognize the end of the target sequence. The newly formed strands have a beginning which is precisely defined 5 days end of the primer. Applications of PCR. It is a reliable method for detecting all manner of mutations associated with genetic disease. Duchenne muscular dystrophy is an example of genetic disease whose detection has been greatly simplified by the use of PCR. Identify bacteria and viruses. The diagnosis of AIDS. PCR can be used to detect a small percentage of cells infected by the human immunodeficiency virus. This is important in cases where there are not enough DNA available for research, such as at a crime scene or identification of ancient species. RT-PCR. It is the most sensitive techniques for mRNA detection. Commonly used techniques for quantifying mRNA levels. Northern blot analysis and RNase protection assay and RT-PCR can be used to quantify mRNA levels from much smaller samples. RT-PCR technique is same like a PCR techniques, but it uses a reverse transcriptase enzyme to convert RNA to copy DNA, followed by PCR to amplify the DNA. RT-PCR is used to locate and quantify known sequence of mRNA in a sample. The first step in RT-PCR uses reverse transcriptase and a primer to anneal and extend a desired mRNA sequence. If the mRNA is present, the reverse transcriptase and primer will anneal to the mRNA sequence and transcribe a complementary strand of DNA. This strand is then replicated with primer and check polymerase, and PCR protocol is followed smaller amount of time to produce a significant amount of copied DNA. Applications of RT-PCR. Used in genetic studies that allow the detection and quantification of mRNA that shows whether or not a specific gene is being expressed in a given sample. RT-PCR is a very important test in the field of genetically modified organisms because it gives researchers a mechanism to test whether any specific gene is turned on or turned off.